So I think that it's time to start. We are going to pick up the polar seminars within the research seminars of the National Natural Science Museum in Madrid. So now these seminars is called Seminars Andrés Barbosa in honor of our colleague. So today is going to be the first seminar of this cycle, and it's going to be presented by María Vila Costa. She is a senior scientist at the Institute of Environmental Assessment and Water Research from CSIC. She did her PhD thesis at Marine Science Institute, also from CSIC in Barcelona, on the sulfur cycle in the oceans and the role of the microorganisms. Later, she did a postdoc in the laboratory of Mary Ann Moran at Georgia University in the United States as Marie Curie. She returned to Spain with a Juan de la Cierva contract, specifically in the Center of Advanced Studies of Blanes, also in CSIC, where she worked on high mountain lakes, and joined later to the IDAEA, where it's actually as a Ramón y Cajal. I would like to highlight her polar research trajectory through participation and co-leadership of project, direction of students, states, and so on. Not only in Antarctica, also in the Arctic ones. So today, she's going to present her last studies about capacities of polar marine microorganisms for the degradation of organic pollutants. I think that we have bad news because we have these pollutants also in polar ecosystem, but good news because it seems that microorganisms can help us to degrade them. So it's your time.
Thank you very much, Asun, for this nice introduction. And a little bit of a spoil. Now you say the microorganisms play a significant role. Thank you very much for inviting me also to give this talk today, and I'm very happy to be able to speak a little bit about marine microorganisms and organic pollutants in Antarctica. So when the general audience think on ocean pollution, they imagine what you see here, an ocean full of plastics, debris, oil spills, right? But the truth is that most of the pollution of our oceans is accounted by hundreds of thousands of different compounds. Many of them are organic pollutants. They are synthetic compounds. Many of them are organic pollutants that are actually components of our daily life products, like these perfluoroalkyl substances that they are called as forever chemicals. Their persistency, you can see they can be found in all the oceans, including Arctic and Antarctica, and they are components of the Teflon, these Gore-Tex jackets we all wear when we go to the mountains, or the boots. These are part of them. Or another family of emerging pollutants of environmental concern, the organophosphorates, the flame retardants, and plasticizers, that are components, for instance, of the seats where you are sitting right now. They also reach Arctic and Antarctica, and also all the oceans, and they have this widespread distribution. Or historical pollutants that, due to their persistency and toxicity, they've been banned to be produced in the last 50 years, like the PCBs and the HCBs, and you can still find them in your samples when you take it now and you analyze. Or, combustion subproducts like the dioxins, the furans, the hydrocarbons, aliphatics, aromatics, obviously plastics, microplastics, nanoplastics, and all of these compounds, plus hundreds of others that we don't have the analytical techniques to identify and quantify each of them, compose a complex mixture that we call anthropogenic dissolved organic matter that enter into the cycle of elements like any other biogenic carbon compound produced by phytoplankton, here in green in this line, that it's cycled through the microbial loop. These compounds, they are produced on land, they can be released, just run off directly from the continents to the ocean, or undergo atmospheric transport, be deposited into the oceans, and once in the water column, they just go through the. But they have one difference if we compare to the natural organic compounds. And the main difference is these characteristics. They are highly hydrophobic, so they have a huge tendency to bioaccumulate into the organic tissues, in the lipids, and since many of them are toxic, they pose threat to ecosystems. In order to illustrate that, I put just here an example. These historical families of pollutants, I told you, they haven't been produced in the last 50 years, the PCBs. The bioaccumulation of PCBs in killer whales, it's forecast that it's going to decrease the population of these animals by a half in the next 50 years. So they have a relevance, right? These bioaccumulated PCBs will originate in the reproductive system, in the immunological system, and so on. No? One can ask, okay, if that happens to the killer whales, what happened to us? But this is another story. We know these pollutants reach the open oceans, Arctic and Antarctic, by atmospheric deposition, but even if I say that, you may ask yourself, okay, this is a lot, this is just a little bit, this is something relevant, something we should be concerned about it. If we take, just to illustrate, one family of these organic pollutants, the polycyclic aromatic hydrocarbons, these are products of the incomplete combustion of organic matter, including fossil fuels. They are toxic, cancerogenic, and so on. You can see that you can find them these are the concentrations in the atmosphere, in the marine atmosphere, in the dissolved phase of the water column, in the particles, because they bioaccumulate. And if we compare the annual entrance of these compounds by atmospheric deposition, that we estimated to be 1 teragram a year, or 400 if we take into account as well those pH that we don't have the standards to quantify each of them, that equals to 4 times the pH released by the worst oil spill accident ever occurred in our oceans that you may remember. It was the Deepwater Horizon oil spill in the Gulf of Mexico in 2010, I think. So in other words, what is arriving by atmospheric deposition is the same that having 52 oil spills in our ocean year after year after year after year. And this is only one family of pollutants. And these ones also reach Arctic and Antarctica. This is all bad news. Well, as Asun said, this is not all bad news. We have some good news. We were able to estimate the main sink of these pollutants in the water column, which is microbial biodegradation, that accounts for 99% of this removal of pH from the water column. But that was calculated. We estimated that in the temperate waters of the ocean. We don't know what happened in the Southern Ocean. We don't know what happened in Antarctica. As I told you, another characteristic of these compounds is that since they are semi-volatile, they can undergo long-range transport temperatures, and this is how they reach, even if they are produced mainly in the Northern Hemisphere, they undergo a series of cycles of volatilization and deposition, depending on the temperature, depending if it's summer or if it's winter, until they reach, it's like a global distillation system, until they reach the polar environment where they get trapped due to the low temperatures. To illustrate this trapping, the concentrations we measure there are much higher than expected given the long distance they are from the sources, right? This is very fresh data from our last campaigns. We did one latitudinal transect from Spain to Punta Arenas, crossing the whole Atlantic, and then so following this latitudinal transect in Antarctica as well to very low latitudes. And this is the concentrations of PFAS, one family of these organic pollutants. And you can see, this is part of the thesis of Nuria Trilla, a PhD student of our group. The concentrations are in the range of those measured in the Northern Hemisphere near the coast, and much higher than those measured in the central gyres or in the middle of the ocean. So this trapping exists. Pollutants are relevant in Antarctica. We know bacteria can degrade them. So our scientific questions were, okay, are pollen microorganisms a significant sink of these compounds in Antarctica? And if so, what are the metabolisms? How do they do it? Extreme environments, we know they promote new metabolisms. Some of them facilitate the degradation of organic pollutants, and we can just, go there, fish them, and identify. In order to answer these questions, we will focus on the experiments we've done in Antarctica that are part of this large collection of experiments we've been doing in the last 10 years in all the oceans, studying this interaction between marine microorganisms and organic pollutants. And from the whole pool, this complex mixture of pollutants, I would like to focus today on 3 main families, the PAH, the PFAS, and the OPs. And we start with the PAH that I presented before. These molecules, and you can see here, are composed by multiple fused aromatic rings. Depending on the number of rings, will be high molecular weight or low molecular weight. They are toxic, cancerogenic, and so on. And we are lucky in this case because this family of pollutants is the best characterized family of pollutants, thanks mainly to all the research done on oil spill situations. When there is an oil spill, everyone looks for the scientists to see how do we solve that. And there is a lot of research done on bioremediation. And this is why we know which bacteria consume pH in seawater. They are called hydrocarbonoclastic bacteria, that they can be facultative or obligate degraders. And we also know all the degradation routes from the molecule to their respiration and each of the genes that encode for each of these steps of degradation. This is a lot of information. We don't have this information for any of the other families of pollutants. So the first question we had was, okay, we go to Antarctica, there is any correlation between the in situ concentrations of these hydrocarbonoclastic bacteria and the in situ concentrations of pH in seawater? In order to answer that question, we collected data from 3 different campaigns. We did in 2014, 2017, and 2018 in the coastal waters of the 2 Spanish base stations, Juan Carlos I in Livingston Island, Gabriel de Castilla in Deception Island. Both are located in the South Shetland Islands, very near to the Antarctic Peninsula. This is, what you possibly know, right? In the site of Argentina and Chile. And we went then during the austral summer. We did the sampling of the water, the sampling of the plankton. We collected the pollutants. We also collected the biomass. From this biomass, we extracted the DNA, and we did 16S amplicon sequencing. That is the gene that allows you for the taxonomical characterization of the community. If you want to know what's the community composition of a microbial community, you do a 16S amplicon sequencing. This is the initial data. This is the austral summers, all the days in the Livingston 2014, Livingston 2017, and Deception Island 2016. The concentrations of pH in the dissolved phase in blue, low molecular weight pH, here are named them, and the high molecular weight in red. And actually, the only thing I would like you to focus today is look at this red line. This is the seawater temperature. And as you can see, here in Livingston Island 2014, when the temperature was increasing, the concentrations of pH in seawater were increasing. But that didn't happen in the other 2 campaigns. Why is that? The answer, you can see it in these 2 pictures. And if anyone wants to guess, I give the answer correctly. There's no. Very correct. In the campaign in 2015, there was a huge amount of snowpacks, on the continent, while in 2018 and 2017 in Deception Island, there was no snow. So that makes us hypothesize that when there is snow during this austral summer rise of temperatures, this snow is melting, giving an input of pH into seawater. So we next analyzed the geochemical markers of degradation, of biodegradation of these compounds, and that will be too long to explain, but the only thing I would like you to see is, again, this is the austral summer and the different campaigns. There is a very different pattern between Livingston Island 2018 and 17 in Deception Island and 2015 in Livingston Island, suggesting there was more biodegradation when the concentrations of pH in seawater were higher. Then we analyzed the microbial community composition. Here you have, again, the days of the austral summer. This is the relative contribution of the different taxa. This is just the general composition we observe in Antarctica. And the interesting thing I would like you to focus today is that when we look for correlations, this is a heat map of correlations between the relative abundance of the different taxa and environmental parameters, including pH concentrations. In red, when the correlation is positive, in blue, when it's negative, with a star when this correlation is significant. Those bacteria that they belong to the hydrocarbonoclastic ones were positively correlated to the concentrations of pH, suggesting that they do play a role on the fate of this pH in seawater. So when there is more snow, there is more biodegradation because there is more concentrations of pH. And then we ask ourselves, okay, look, this is the Antarctic ecosystem. Where of this graph there is the highest concentrations of pollutants? We know in the snow there are, but they actually, when they reach seawater, is when these concentrations are really high. We know there is atmospheric deposition of these pollutants, and some pollutants can also reach Antarctica through sea spray aerosols. And the answer, and this is not exclusively for Antarctica, only in the high latitude lakes happens, the main collector of any atmospheric deposition is what we call the surface microlayer, is what we call the skin of the ocean, the few millimeters on the top of the water column that not only collect the pollutants that fall from the atmosphere or that get deposited from the atmosphere, but also since they are super hydrophobic, they get enriched from subsurface waters. So this layer, and it's been published before by us and by other people, they have a higher concentrations of pollutants. And we know there is this bacterial neustone. There are bacteria that live there, that they must be used to live not only under high concentrations of pollutants, but also under really hard conditions, because the UV radiation is really hard here. You know, there is many events that make the life really hard. So our next question was, are these bacteria more prone to degrade pH? First, they are more abundant, and if they are, do they efficiently consume organic pollutants? Here you can see Alicia and Gemma sampling this surface microlayer with a glass plate. This is in Livingston Island. And the first paper we published is a bit of a spoiling because already says that, yes, the concentration, you know, there is an enrichment of organic pollutant degraders in the surface microlayer. But let's look at the data. When we did the most basic analysis you can do in a bacterial community, which is analyzing the bacterial abundance by flow cytometry, systematically, here is the Austral summer of the campaign in 2018 in Livingston Island, and this is just the concentrations of bacteria in the samples. Systematically, the concentration, the abundance of bacteria in the surface microlayer was higher than in the subsurface waters. And when we look at the composition of these communities, in this heat map represents the ratio between the relative abundance of the different taxa in the different days in the surface microlayer versus the subsurface waters, in red when they are more abundant in the microlayer, in blue when they are more abundant in the subsurface waters. If you can see, the most red tiles we can see in this heat map belong to hydrocarbonoclastic bacteria, the pH degraders. Okay, they are there, they are more abundant, but do they also consume better this pH? We went again in the same campaign, we collect exactly the same way the water, and in this case, we did experiments. And now I would like to do a point here, because when we do these exposure experiments that we expose the communities, the microbial communities to pollutants, we don't do experiments where we want to see who survive. We do experiments from a biogeochemical point of view. We add really low concentrations of pollutants. We don't want to stimulate new metabolisms. We don't want to see what happens when the concentrations of pollutants are extreme, like in toxicological studies. We don't do toxicology, we do biogeochemistry. So two characteristics of our experiment: concentrations are within the range of their environmental variability, and two are very short, 24 hours, maximum 48. We get super wild if we do 48 hours. And then in this case, what we did was collecting water from the surface microlayer, water from the subsurface waters, two controls where we didn't add any compound, and the pH exposed communities or incubations. And what did we observe? This is the pH turnover rate. In this case, you can see the different compounds. We put it in order from less hydrophobic to more hydrophobic. That is also the lower molecular weight and higher molecular weight. This is the turnover rate. This is the difference between concentrations at the final time point minus initial time point corrected by time. So it's the speed of degradation. As you can see in blue, surface microlayer, in red, in green, the subsurface waters. In the controls, there was no degradation of pH. But in the pH-exposed communities, we observe a significant removal of those pH of high molecular weight, which was really surprising. These are the most refractory ones, the most resistant to be degraded. But our biological data supported fully these results. We also did this 16S sequencing of the community, characterize the community composition. In this case, we distinguish between the free living fraction and the particle attached. And very briefly, I don't want you to look at everything. This is the different taxa. This heat map represents relative abundance of each of these groups in the different treatments: surface microlayer, subsurface water, free living, particle attached, control, and pH enrichment. In blue is higher abundance, higher relative contributions. In yellow, less. When there is a letter here, indicates a significant difference between abundance between treatments and controls. And the only thing I would like you to focus today is to see here, this square here. These are the hydrocarbonoclastic bacteria that some of them, like the Pseudothermonas, increase by 4 their relative contribution to the community, which is unbelievable under these low concentrations, low temperatures that you find in Antarctica, and only in 24 hours. And surprisingly or not, this is very similar to what is observed when there is an oil spill, and when the concentrations of pH are super, super huge. And this has been attributed to their capacity to exudate exopolysaccharide substances that act as biosurfactants and facilitate the biodegradation of these compounds. Don't worry, this is the metatranscriptomics. This is a snapshot of the gene expression profile of the communities, what bacteria were doing at that moment. And the only thing I would like you to focus today, these are the different metabolisms of the cells. These are the different taxa. In red, when they were enriched in the pH treatment versus the control. Here, the numbers indicate those genes that they were significantly expressed in the pH treatment versus the controls, when the difference of these transcripts were significant. And the only thing I would like you to see today is that most of the activity happened in the surface microlayer, free living fraction, when we spike them with pH. But if we look exclusively those genes encoding for the pH degradation, maximum expression was happening in the subsurface water particle microbiome. Just to summarize, we observed degradation in the surface microlayer. In the particle attached fraction of the surface microlayer, community already changed in 24 hours, indicating a super, super fast capacity to degrade pH. But the transcriptomical activities were higher in the free living fraction and also in the particle attached of subsurface waters, suggesting that this particle microbiome should be investigated more in depth in the future regarding their capacity to degrade pollutants. So, okay, pH, yes, there is degradation in Antarctica. What about these PFAS? The PFAS, also known as forever chemicals, that give you an idea how persistent they are in the environment. They are molecules that are composed with these fluorinated carbon chains and different polar heads. When this polar head is a sulfonate, it's the PFOS. When it's an acid, it's a PFOA. There are different lengths of the carbon chain, but this with 8 carbons is the most common one. They are toxic, they are carcinogenic. That was a big, big, big concern when they were found in the drinking water and their effects in the babies when, pregnant women. They have many, 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 many harmful effects for health. But they confer surfactant properties to the products where they are added that makes them super good for anti-adherent, water repellent surface and so on. This is the Cortex component. And if we look at the ratio of concentration between PFOS and PFOA, these two very abundant PFOS compounds, look, in temperate waters, the ratio is around 4, but in Antarctica is around 1. Why is that? It's because PFOS don't reach there, or it's because there is a biodegradation of PFOS there? And this is actually a super wild question to ask. If you were in the field, you were like, what? I mean, it's so stupid to ask yourself if there is any biodegradation of PFOS, because there is no reports that so far until that moment that report degradation of PFOS. They just don't degrade. But we say, okay, we go to Antarctica, let's do experiments. And let's see if these forever chemicals, they can be degraded. So in this case, we did the campaign in Gabriel de Castilla. This is in Deception Island. It's a bay with volcanic activity still. underwater. Here you can see Elena sampling her water for the experiment, carrying to the lab, and here the exposure experiments. And again, we have the control, and we have the addition of PFOS and PFOA. And how surprising was to see that the PFOS concentration decreased only in 24 hours in our samples when we spike these PFOS. And we were lucky enough to collect samples to look for the gene expression profiles of these communities after 24 hours. And if we look at the different metabolisms of the cells, in blue, the controls, in red, the spike incubations, these are the relative abundance. The stars indicate when this difference between treatments and controls is significant. You observe that there was only difference in the DNA metabolism. That always happens. It's not so important. But the sulfur metabolism was significantly enriched. And if we zoom and we look which metabolites were those, they were related to the DMSP consumption. DMSP, you may know it, is the dimethyl sulfur propionate. I did my PhD on that, so imagine how surprised I was to see, oh, I mean, this is the cosmos telling me something. And they were related to the sulfur oxidation and sulfur consumption, and the layers of the carbon and sulfur bond. So we did, this is not very polite, but a Chinese work, like looking each of the genes that they were significantly differently expressed between treatments and controls, and we were able to draw and propose an hypothetical pathway of degradation of these PFOS that was actually a desulfurization of the molecule, and bacteria are actually using this ionic head, the sulfonate, just as a source of sulfur for the amino acids and proteins, or just as a reducing power, as they do with DMSP. And nowadays, we are working on cultures from those experiments to try to get a more simple system, not this complex mixture of many taxa, but only one species of bacteria to elucidate the degradation pathways and the degradation products, which is a really interesting issue. Okay, PFOS, yeah, we were able to show that they were degraded. And finally, the OPEs, the last family I would like to talk today. These organophosphate esters, flame retardants, and plasticizers, the organophosphate esters may be familiar to you because it's like the mustard gas that it was used in the First World War, el gas mostaza. Or there are many pesticides that they are also organophosphate esters, but those ones are really labeled. They can be biodegraded quite easily. The ones that they are persistent enough to reach Arctic and Antarctica are the pollutants and plasticizers. That they have this structure. You have the phosphate esters, and then you have different radicals that they can be aromatic, they can be halogenated, they can be just, a carbon chain. In this review we did this year, we reported all the OPE concentrations in all over the oceans, reported so far in the literature. And as you can see, in the Southern Ocean is very few. So that project, Antom, that was led by Jordi Dax and Begoña Jiménez, that consisted in 2 cruises, the ones I told before, the latitudinal transect from Spain to Punta Arenas and in Antarctica, was mainly focused on measuring concentrations of OPEs, turnover, fates, and so on. Here I put you some pictures to see how do work there in the lab, sampling the water, going for the microlayer. We did also some outreach. We are presenting results in different meetings right now. But the question I would like to show you today very, very briefly is that one that we ask ourselves. These OPEs that they have phosphorus, and phosphorus is a limiting nutrient in the oceans, is actually this artificial one, this synthetic one, anthropogenic one, is it used as a source of phosphorus or carbon by marine bacteria in Antarctica? And in order to do that, in this case, what we did was enrichment experiments, and we worked with cultures. Once in Barcelona, in the lab, we incubated these cultures that we could isolate after enriching the seawater from Antarctica with OPEs, with artificial seawater limited by carbon, and artificial seawater limited by phosphorus. Then we add the OPEs, then we add the different strains we have, And then we did the sampling of the analysis of the growth curves and the consumption rates. Just to point that we have developed a new methodology to measure these OPEs in the cultures that replace the super tedious and time-consuming procedures we have to follow when it's just seawater sampling. This is part of the postdoc we have in the lab. Is a Santh in working with Nayara Berrojalviz, that those of you that have been in Antarctica, you possibly have shared Antarctic experience with her. And the results, here in violet, you see the incubation with OPE without any carbon source, or OPE without any phosphorus source in green. This is the difference between concentrations when there is bacteria and when there is no bacteria, indicating that anything below this line indicates consumption. And this is, I only selected examples of two nice ones that they consume OPEs. On the top, they consume OPEs when they don't have carbon or they don't have phosphorus. The one below, they consume OPEs when they don't have phosphorus. And this is more or less also reflected in the growth curves, more or less because it's not super precise, or what you will expect if you are actually doping your strains with a super yummy substrate like glucose or so on. But we observe this consumption and we observe difference in the growth. And what we are doing nowadays with this new project is trying to use these strains to inoculate communities in bacterial electrochemical systems. This is not microbial fuel cells. This is the next step. When you, in microbial fuel cells, you apply a current, you create a microbial electrochemical system that promote reactions that spontaneously they wouldn't happen. So our idea behind is that with specific strains, we may be able to build a microbial cell that will enhance the degradation of pollutants in seawater. I will tell you in 5 years if we succeed or not. Okay, so final question we had today. Yes, potentially there is consumption of OPEs, and we are waiting for more data from the last campaigns to really answer, yes, there is OPE consumption in Antarctica. And this is just the project we are now, also with Jordi Dax, with Begoña Jiménez, and members of the lab, that it's called PANTOC. And again, we go this winter in Livingston Island and Gabriel de Castilla, and we are widening our range of pollutants that we measure there and their interaction with microbial communities. And I would like to do a lot of publicity of the Instagram account that the students of our lab have created. They accumulate more than 400 followers now, I think almost 500. And they are doing a really amazing job explaining things about Antarctica, why we go there, and which kind of research is carried out in these sites. We will do what we've been doing, sampling different matrices, using the different techniques I show you along this talk, including lake water and rain and snow. And as example of this super, today they told me I have to say hype reel, which translated means a super cool short video in the Instagram language. And I really encourage you to be followers.
Well, yes, we are going to Antarctica, but no, we are not going on a trip. Come here, let me show you. Running down to the riptide, taken away to the dark side. I want to be your left-hand man. I love you when you're singing that song, and I got a lump in my throat 'cause you're gonna sing the words wrong.
Yeah. And obviously, this is not only my work; it's a teamwork. And this is our research group in IDAEA in Barcelona. And thank you very much for your attention.
Thanks, Maria, for your interesting talk. So now we have time for questions. No one? So perhaps I can start. I would like to ask, because you have been talking about bacteria and also a bit of archaea, but you have not said anything about fungi. It is possible that also they are involved in some of this process?
Possibly. We haven't looked for it. And now there is this emerging interest on fungi, at least in the water column. I mean, I know on land it's been already. And yeah, possibly they play a role as well.
Okay. And also another question, because sometimes I have seen that you have collected also soils and different. So have you made any analysis also with microorganisms from the soils?
The only attempt I've done at the beginning of my Ramon y Cajal was looking for antibiotic-resistant genes, but it was a big failure. They were not there. Because we have a time series of 10 years of soil collected in the stations. Every time we were going, we were collecting and freezing. But there was, I thought that I would see a tendency, an increase, but no. They were
okay, no, but perhaps in any moment we can check, because also we have found sulfur metabolisms in the soils.
Then, yeah, sure.
Okay.
Thank you.
There is a question? Do permanent research base contaminate with organic substance derived from the petroleum, plastic, and fuel oil? Your ships' engines also polluted the waters near the research station?
Yes. Actually, there is a very recent publication that is reporting microplastics in the sediments, that they are more abundant in summer when the Antarctic base stations are open. But that impact, I mean, it's even higher due to the tourism, that it's only increasing in Antarctica, and there is more and more and more. So there is more research to see if, this is a pattern that we can confirm. There is more pollution when there is more tourism, in order to legislate or just take decisions regarding that. It's not only the base stations, it's mainly the tourism.
Okay. Also, we have congratulations for Fernando Bollo. They say many thanks for your interesting and well-supported talk and research. Congratulations to you and your colleagues of PANTOC.
Thank you very much.
Rafa?
Thank you so much for the nice talk. I have two questions. You have shown transcriptomic data, but I'm not sure if you were mapping the transcripts to metagenomes or to metagenome assembled genomes, so you were assembling the transcripts. That's the first question. And the second question, I don't remember when, but at some point you showed that the particle fraction has more activity. And I was wondering if that could be related to microplastics. Maybe to that those genes, although the pathways are characterized, it's not super clear which are the substrates at the end, if those genes could be related to microplastic, if you have checked on that.
Good point. The first one is a very good question, because the field of bioinformatics change every second. When we did these experiments, we did assembly-based annotation, and we compared results to the read-based annotation. Nowadays, we do assembly-based, and then we do mapping to GTDB genomes. And we are using different pipelines that, what we always do is we try 2 or 3, and when the results are consistent, then we take it for good. And I can tell you the ones we use. And that's the first question. And the second one, absolutely yes. Actually, the plastics are like passive samples of pollutants. They attract pollutants while they are also polluting. I mean, it's a bit crazy, but it's like this. And the plastisphere is this, biofilm that is growing there, that actually in this coming campaign in PANTOC, we have one PhD student exclusively, her PhD is focused on studying that. So I will tell you in a couple of years.
Thank you for the talk.
Thank you.
I think I was together with people from your team anytime in Deception Island, and I think they were sampling in the inner bay of Deception and also in the penguin area,
yes,
in the lakes, and I don't know if maybe in the outside. And I was wondering if there was any difference in the concentration of bacteria.
Right. Here the point is that when they were sampling the penguin and the water, they were not doing experiments with that. It was mostly like measuring concentrations and characterization of the communities. But we don't know anything about in situ activity because we didn't incubate the waters. It could be interesting to do it. Maybe this year when they go back, they can do experiments with lakes. Yeah. Thank you.
Thank you very much for your talk. I really liked it. And even knowing that I'm not related to biochemistry, and I think I got everything, so really, it was really nice. But I have two questions. The first one is, you presented results for 3 bacterial families. And for the first one, you made a cultive, and you said, I think it was in the image, it was you cultivated in the dark.
Yes.
But that cultive in the micro layer, it isn't in the dark.
No.
Okay, so wouldn't it affect the results of the?
But if you remember, the incubations are super short. They are 24 hours. If you add the light factor, you are increasing the complexity of the experiment, because then you have the phytoplankton producing, just doing the photosynthesis, normal, but then they exudate organic matter, and then there is an interference, and then you need to multiply the number of bottles you do this incubation, because I didn't say that, but logistically this is super challenging, because we cannot use the same bottles for chemical analysis and biological analysis, so everything goes in, replicates in parallel, and only these experiments were 36 bottles being incubated there. And if we did include solar radiation in Anton experiments, because we were on the ship, we were in Desperides, and it was a bit easier, to incubate the bottles, but I still don't have the results. But you have to do a lot of replicates to really make sure your results are correct. Yeah.
Okay, thank you. And the second question is related to that same family of bacteria. Why you said that they are more known in academic? So why is it?
The hydrocarbonoclastic bacteria, because these are the ones that clean the ocean when there is an oil spill. When there is an oil spill, these hydrocarbons, it's this image, chapapote that we see, and these are the bacteria that immediately respond and start cleaning. And what they found is that since they are producing these surfactants, these biosurfactants, what they make is they take the petroleum and they make little bubbles, and with little bubbles they consume even better, and then the bioremediation is enhanced. And what they observed in these experiments, in the experiment in the Gulf of Mexico, when there was this super huge oil accident, was that from the planes, if they were throwing surfactants, they thought they were enhancing biodegradation, but actually they were chemical surfactants. So they were killing the bacteria, these hydrocarbonoclastic bacteria. And now they use biosurfactants, very, very similar to the ones they produce. So it's this kind of knowledge that they build in order to improve bioremediation. Yeah.
Thank you.
I'm not from this field at all. I'm from glaciology, but I wonder whether when you do your analysis based on particles or concentration of particles collected on the water near coastal areas, do you take into account or correct for the fact that you have mentioned with your example of I think it was, I can't remember the numbers, but they were 2016, 17 as compared to 2000, maybe 14?
14, yeah.
14, that was a year without any snow melt. Just to correct, because this has not been something exceptional, but we had in this area between 2009 and 2015-16, really low temperatures, really low snow melt, and so you had this situation. So do you, in your analysis, correct for the fact of how much snow has melted and then the water has taken this higher or not, this higher concentration of particles?
It's difficult to correct it because the salinity doesn't change, actually. Even if the snow melt is an important input, it doesn't change the salinity of the coastal water. So it's difficult to correct for that. What the thing we could kind of correct for was if there is a lot of particles, number of particles, that it may be also related to what you are questioning, and size fraction We cannot characterize which particles we have in each sample. But by size fractionation, we try to select, and just subsample.
No, but perhaps in case you had some years where you had samples and different amounts of snow melting and data between both, maybe you could try to find some kind of correlation that could help you to make
Absolutely, and to see, and to see how many
but
particles.
I don't know the range of years where you have your samples and the available snow.
It's something we could look at it. Yes, absolutely. Just by the instruments we have, with the flow cytometer, you don't see that because it's a. But yes, absolutely, yes. Thank you.
We have another question. Thank you for the talk. You mentioned the bioaccumulation of organic pollutants, and I would like to know if whether plastic or microplastic might also serve as carrier for these pollutants.
Yes, they do. They are carriers of this. Well, there is this big discussion in the field if plastics are more like a source or an absorbent of pollutants, because they are both, is what we said before. They can absorb pollutants and then carry them, but they also, because they get old, while the longer they stay in the ocean, the more they lixiviate, all these additives they contain, and the additives are actually really toxic ones, not only the polymeric side. And it's an open question, what are more, if they are just a source of toxicity by themselves, by the pollutants they absorb. But definitely is what they are. They are a source of pollution. Okay.
So no more question online. So I don't know if there is any question more here. So the last one.
Thank you, Maria. As far as I know, normally the degradation is higher when the temperatures are higher. But here we have very low temperatures.
Yes.
So I was wondering if this is because we have this degradation that you detect is because the temperatures are very low and this is because there are very specific microorganisms, secrophiles doing
Correct.
this, or perhaps those microbes are everywhere, and if you decrease the temperatures, I don't know, in the tropics, you can also have this degradation.
I think it's the first option you said, is because these are bacteria, they're really used to live under very low concentrations. And I can tell you that because the work we've done with the strains, we also play with temperatures. And those Antarctic ones, they don't at 30 degrees, while the other way around also happens. When we put them in the fridge, the ones that they come from the Mediterranean, they don't grow. So these are bacteria really used to live under optimal growth under 1, 2, 3, 4 degrees.
Do you think that you can find the same in the North Pole, the same strains?
Absolutely, yeah. I didn't show the results in Arctic because we've been there only once, but the results we found that campaign were very similar to the ones we observed in Antarctica. It's just that I cannot build a story because it was only one time. But
that is very interesting history thing.
Thank you very much.
I think that we are going to finish. Thanks a lot, Maria, for coming here and giving this interesting talk. And we wait you for the next seminar that will be in 27th of October.
Thank you.